Good morning to the dear aspirants who are going to appear for the Pollution Control Board examinations. So, like all the PSC examination, Zodiac is part of your career builders. So, in that sense, we are giving some tips to the candidates or the aspirants to relieve from the huge syllabus published by the PSC in connection with the examination. Here, we have taken another area, plant tissue culture is a part of plant biotechnology. Okay, that will be very familiar to the biology students, not for the chemistry students or any other science. So, that is why we want to make you more simpler how you can go through that part of the chapter, namely plant animal biotechnology. Okay. So, here we are going to shoot out some questions randomly, maybe the possible question for your PSC examination. Okay. So, we will see how the questions can be shooted out. The first question is that which of the following plant cells show totipotency? As I said earlier, if you are a biology student, he or she can immediately answer, because the word totipotency is very important for plant tissue culture. That's which means potentiality of the cell to undergo rapid multiplication. Okay. So, it is a potentiality of the living cell capable of undergoing divisions, what we commonly call totipotency. It is a quality of plant cells, not commonly occur in animal cells except certain fibroblast cells. Okay. So, here out of the given options, cork cells, dead cells, they are completely dead, non-living. So, no way of answer. Meristem, living, immature, actively dividing cell, correct answer. Sieve tube, cell without nucleus, so unable to divide, so not the answer. Xylem vessels, non-living functional cell, not the answer. So, the answer is option B. Meristems are unique in the sense that they are immature, actively dividing cells. That means they are capable of doing totipotency property. But that is the answer. Okay. But this is one of the simplest way of question in the tissue culture part. Second one is, what is callus? Again, a very basic question, because in your syllabus the word is there, callus. When I put the word callus, which is denoting immature, unorganized mass of cells capable of division. Okay, that is the callus, immature or unorganized mass of cells capable of division. So, whenever you are culturing in explants or a part of a plant like shoot tip or node or internode or anther, anything across, it produces callus. Okay. But here the answer is tissue that grow to form a embryoid, an unorganized activity dividing the mass of cells maintained in a culture, an insoluble carbohydrates, a tissue that grow from an embryo. The answer is option B, because they are unorganized cells or tissue capable of undergoing division in a culture vessel or in a culture medium. That is what we commonly put the word callus. Okay. But it is a prerequisite for developing a cell into an in vitro plant. Okay. That is another way of question. Basic, simple question. Question number 3. So, that is slightly tough. Which of the following chemicals are mostly widely used for protoplasmic fusion or protoplast fusion? Because nowadays tissue culture is flourishing like anything. So, we can make the two protoplast of a cell to fuse. Okay, we can make the two protoplast of a cell to fuse, so that you are getting some more combination of characters. What are the characters occurring in the protoplasm can be made into a single body. For that we are using a chemical. So, which chemical is commonly used or facilitating for or enhancing the fusion of protoplasm? So, what given example is mannitol. It is a sugar alcohol, not the answer. Polyethylene glycol, what we commonly call PEG, that is the answer. It is a non-reactive, non-toxic, higher molecular weight compound capable of protoplasmic fusion. Correct answer. Sorbitol, wrong. Mannose, wrong. So, they are mere chemicals, no way to role play in tissue cultures. So, the correct answer here is PEG or polyethylene glycol, which cause the event called fusion between protoplasm of cells. Okay, but that is another question. Again, slightly in continuation, I place the question here. Cybrid are produced by, there is a clue in the question, cybrid. So, that means fusion between cytoplasm. Okay, but nucleus from a cell only, because every cell have got nucleus, every cell has got cytoplasm. Here we are using the fusion between two cytoplasm of two cells, but you are taking nucleus from a single cell, other will be discarded. Okay, that is the question here. So, the answer is the nucleus of one species, but cytoplasm from both the parental species. Correct answer. The fusion between two same nuclei from the same species, wrong. The fusion of two different nuclei from different species, wrong. The fusion of two same nuclei from the different species, wrong. So, it is a fusion between two cytoplasm, but we will take only one nucleus of a cell, other will be discarded or eliminated. Then you are placing a word called cybrid. So, some of the tissue culture work we are requiring cybrids, that is the answer. Then normally you know that based upon the chromosome number, we call the cells either by the term haploid or by the term diploid. Okay. So, haploid is commonly occurring in the gametangel cells, for example, male gamete, female gamete, etc. The plant body is usually or animal body is usually diploid. Okay. Here in tissue culture, in some of the area we are requiring haploid plants, because we want to transfer some ideal qualities of a male or a female gametic cells. Okay. Haploid plants can be obtained from, one is anther culture, second is bud culture, and third is leaf culture, and fourth is root culture. When I put the word leaf, bud or root, all are diploid because they are part of the plants, but anther they producing the male gametic cell in future. So, the correct answer is we prefer anther, or you can put the answer ovule culture, but not in the option. Okay, but here the answer is anther culture, because anther culture can produce pollen grain or microspores. From there you can produce haploid plants. Okay, that is another way of question. So, again nowadays we are going for producing synthetic seeds. Normally a seed have got an embryo covered by a seed coat. Seed coat is a barrier or a protector of the embryo. Okay. But here the concept here is that you are producing an embryo from tissue culture and you are encapsulating the embryo with artificial seed coat. Then we place the word called synthetic seeds, artificial seeds. Embryo is live, completely encapsulated by an artificial seed coat using a chemical, which chemical function as seed coat ensheathing the embryo in tissue culture experiments. That was the question. Okay. The answer is sodium alginate. Again, the compound is non-reactive, non-toxic, very important. It is not reactive, non-toxic, but capable of functioning as ideal inert barrier protecting the embryo from damage, and it is biodegradable. When you are putting the synthetic seed in the soil, sodium alginate dissolve out, embryo will germinate. Oh, that is another mode of question. Okay. Answer is very simple, sodium alginate. So, again there is another chemical is also given, because since you are going for pollution control board, I made some question from the chemical compounds. Where commonly dimethyl sulfur oxide is used? Because currently a kind of conservation practice is carried at global scale. Okay. For example, our National Bureau of Plant Genetic Resources conserving lakhs and lakhs of seeds for future need without any damage by a mechanism called cryopreservation. The word cryopreservation means preserving part of an organism in a temperature called minus 196 degrees centigrade. You can imagine minus 196 degrees centigrade. So, lowest order of the temperature. Okay. So, when you are keeping the cell or the embryo or the seed there, the cell will break because freezing lower temperature. In order to protect the cell from cryopreservation, we will pre-treat the cell with a chemical what we commonly called dimethyl sulfoxide, so that function as cryoprotectant, because it providing freeze tolerance to the cells. Okay, what we commonly call cryoprotectant, otherwise the cell will damaged. Again remember that this chemical is non-reactive, non-toxic. Okay, like the what I said, sodium alginate, non-reactive, non-toxic, but function as a barrier to protect the cell from freezing lower orders of temperature. That is another way. Then again the very basic question, who is known as the father of tissue culture? Because your title of the topic is plant tissue culture. So, that is maybe. So, who was branded by the term the father of tissue culture? The answer is very simple, Haberland. He is a man. Charles Darwin is origin of species. Murashi is a medium used in tissue culture universally. Gomberg, another medium used in tissue culture universally. Okay. So, the answer is invariably Haberland, known by the term father of tissue culture, because he has done tissue culture in carrots. Okay. Succeeded in the career. So, this is another one, little bit tougher. What are somaclones? Okay. You know the word clone, normally used in plant breeding people or used in farmers, clone, C-L-O-N-E-S. But here the question is somaclones. That is little bit tougher question. Okay, what are they? And here they are given the copy answers, plants which are chemically identical to the source plant, chemically. Plants which are morphologically similar to the original plant. Plants which are anatomically identical to the original plants. Plants which are genetically identical to the source plants. Okay. And I have given the explanation because it is a tougher area. there are various copies of plant tissue culture which are similar to the original plant by which they were grown, and they are called somaclones. So that means they are genetically identical to the source plant. That's why by tissue culture you are producing multiple copies. All the plants are xerox of the mother plant. Okay, like what we are taking xerox copies. So tissue culture is a mechanism by which you are producing multiple plants, or what we commonly call micropropagation, identical genetically, are similar in their look, everything, xerox copies of mother plants. So that what we commonly call somaclones. Clone means, what I said, the first word clone means a product of artificial asexual reproduction. So that's why the word is somaclones are different from the clones, since clones are produced by asexual reproduction. For example, budding you are doing in your house or in the nurseries is an example for clone, asexual reproduction. Somaclones are produced by tissue culture. They are multiple copies of the mother plant which are genetically similar. That's another way of question. So this is a very, very good question. Actually there are 4 questions, I have made it into one. So here I have given some statements. Which among them are truly matched? Okay. So I given option 2, 3, 4, 2, 1, 2, 3, etc., etc. So there are 5 statements are given. Which of the statements are correctly matched? Okay, that was a question here. So it can be 5 different questions again. So I have given the component 1, White's medium is one of the medium universally used. This medium is meant for culturing root cultures, because some of the metabolites medicinally important are produced by the roots. So we do not want the entire plant to culture, or we are culturing only the root. So the statement is correct. MS medium. So already I said the Murash and Skoog, the 2 people made a medium called MS medium, used for organogenesis. That's also true. So you can develop root, stem, leaf or anything across by using the MS medium. Answer is correct. Number 3, B5 medium. So Gomberg medium, what I said the word, B5 medium is used for callus culture, cell suspension culture, protoplasm culture. That's also true. So for doing protoplasm culture, cell culture, or you can go for callus culture, the best ideal medium is B5 medium. There is another medium called N6 medium that's normally used for culturing anther, haploid plant production. Haploid plant production, anther culture, where the media preferred is N6 medium. The last one is niche medium. That's also a substitute for anther culture, because anther culture depends upon the plants or material. Preferable will be N6 or niche medium. So all the 5 are correct, correctly matched. So the option is D, all the correct. Another way of questions. See, once you know the basic, not in depth, you can answer easily any component from the areas. Again a very, very simple question. The culturing of cell in liquid agitated medium is called, because you can culture the medium in solid medium, liquid agitated medium. So we will put a simple name for the liquid agitated medium by the word called suspension culture, because whenever you are culturing a material or an explant in a liquid medium, in a shaker, which normally shows some shaking, agitation with a particular speed, variable again. So normally you know that the speed was referred in terms of rotation per minute, RPM, Okay. So, liquid medium with shaking meant for the program called suspension cultures. Okay, but there is another way of question. Then which of the following is the best suited for method for production of virus free plants? Because normally plants are infected by bacteria, fungus and virus. Okay. But here one of the technique by which we are preferring tissue culture to make virus free plants, because never a virus infecting in tissue culture plants. But here the question is that mode, a very important application: embryo culture, meristem culture, ovule culture, anther culture. So out of that, the most ideal culture which is producing virus free plants, the answer is meristem culture, because meristems are immature actively dividing cells. No virus infect in a meristem cultures. But the answer is option B, another way of questions. Then again, nowadays tissue culture is flourishing like anything, I said earlier, because rather than producing a plant or multiple copies of a plant, now we are going for producing secondary metabolites or drug compounds or Ayurvedic drugs from culturing of a plant cell. See the application. So there is no need for raising a plant or culturing a plant. You can culture a particular compound by culturing a cell or a tissue. So that is the way in tissue culture was flourishing. For culturing a particular metabolite, medicinally important or industrially important, you are adding some triggers or activators, what we commonly call elicitors. Okay. If the question is that, what elicitors are playing the role? The answer is very simple. It triggers the secondary metabolic pathways by which you can produce ideal metabolites. For example, some of the metabolites are universally very important, which can be produced by eliciting the culture media using a chemical. Okay. So many compounds are used. Physical elicitors are there, chemicals are there, biological elicitors are there. Out of that, one can trigger your metabolites. Okay. But the answer is, it is meant for stimulating secondary metabolites. That is the correct answer, option B. Then there is a very, very simple question. Dash is necessary for drying the washed glass vessels, because in tissue culture, aseptic condition is a prerequisite. Whatever you are doing for tissue culture, absolutely free from contaminations or free from any infections. Okay, that is a prerequisite, mandatory. Here the question is, what is necessary for drying the vessels, glass tubes, glass appliances? The answer is, they are given 4 component: vacuum pump, hot air oven, heater, autoclave. The answer is, you are going for removing the drying, so you are going for hot air oven. Hot air oven. So that remove the water content. Okay. So no, don't go for the answer autoclave. Autoclave is meant for sterilizing. Once you want to do the sterilizing component, you go for autoclave. Otherwise, drying is required by hot air oven. So the answer is option B. So again, another question. See the question here, coconut milk and dash are also effective for growth of shoot apices, because coconut milk is a natural hormonal role. That is why currently in tissue culture, they are using coconut milk, what we commonly call coconut water. Okay. So they are given another compound also the same effect. That is synthetic compound. Okay. So gibberellic acid, auxin, cytokinin, ethylene. Okay. Coconut water is a simile to cytokines. Both will facilitate multiplication of shoot apices. Okay. So, if you want to culture a shoot apices, either you can use cytokinin or you can go for coconut water. That is another way of question. So, common man's question. Then again, so I came back to the same component callus. Normally, whenever you are culturing callus, callus will be colored. Some coloration will be seen. So, which component or which pigment yielding the color? Because in plant there are many kind of pigments. One is carotenoid, anthocyanin, chlorophyll, phycocyanin. The callus will be usually orange or brownish in color because of the component called carotenes or carotenoids. Chlorophyll is green, anthocyanin is usually pink or reddish, then phycocyanin is also reddish. So, the color in the callus was fetched by or given by the natural pigment in plants called carotenoids, carrot like colors. Okay, that is a question. So, we are given only some tips, some assistance to you people. So, make use the academy for fulfilling your dream or breaking the barrier of the examinations. So, thank you all, expecting your presence in our classes. Thank you.